Assalamu alaikum everyone. In this video lecture, we will be discussing the topical past papers of the chapter Photosynthesis from A2 Level Biology 9700. This video lecture is the second part of the photosynthesis topical past papers. Let's discuss question number 2. In eukaryotes, the cells of plants and photosynthetic prototists contain chloroplasts. Describe the relationship between the function of chloroplast and its structure. So guys, what we have to do in this question is that we have to link the structure of chloroplast to its function. So what we need to do is that we need to highlight different parts of the chloroplast and we need to relate those parts with the function of the chloroplast. So if we look at the chloroplast, I have drawn a mini chloroplast for you. So we can see that there are different aspects or different parts of the chloroplast and each part is contributing to some function that the chloroplast does. We can see that chloroplast has inner and outer membrane, and these membranes basically allow compartmentalization. And what is meant by compartmentalization? These membranes are separating the processes going on in chloroplast from cytoplasm and from other organelles. This is called compartmentalization. Then we can see starch grains, right? And starch grains over here are produced from the products of photosynthesis. So we all know that in photosynthesis, glucose is made, right? And glucose is stored in the form of starch grain. So the function of starch grain is to store the products of photosynthesis like glucose, right? We can also see circular DNA and 70S ribosomes. These are involved in protein synthesis, right? Circular DNA contains genes which code for proteins that are used by the chloroplast, like the Rubisco enzyme, ATP synthase, and other types of proteins. These are all coded by circular DNA, and these proteins are synthesized at the 70S ribosomes. We know about the stroma, which is the fluid of the chloroplast, and stroma of the chloroplast is the site for light independent stage or Calvin cycle, because stroma contains an enzyme called Rubisco. Then we know about the grana, which are these stacks of thylakoids. The grana contains photosynthetic pigments. These are the sites of the light dependent stage, right? So these are all things that we can write in the question above, that is, describe the relationship between the function of the chloroplast and its structure. So what we can write a lot of points, but you need to know that we just have to write 4 points because the question is of 4 marks. So what we can write is that the first point that we can write is that chloroplast contains grana, right, or thylakoids, which contain what? Which contain photosystems, containing photosynthetic pigments, right? And grana also contains what? Grana and thylakoid also contain electron transport chain, ATP synthase, right? So these all are used in light dependent stage, right? So chloroplast contains grana, thylakoids, which contain photosystems containing photosynthetic pigments, electron transport chain, ATP synthase, all of which are involved in light dependent stage of photosynthesis. So we are relating the structure of the chloroplast with its function, right? This is the first point. Second point is that we can write that stroma is the site of Calvin cycle, which is the light independent stage of photosynthesis, because it contains necessary enzymes and compounds because it contains Rubisco, which is the enzyme required for Calvin cycle, and it also contains RUPP, right, which is required for Calvin cycle because Calvin cycle starts from RUPP. We can also mention that chloroplast has an envelope, which means chloroplast has double membrane, right? Chloroplast has an envelope which allows compartmentalization, All right. We can also mention that there is circular DNA and 70S ribosomes, which are involved in synthesis of proteins, such as, or we can say synthesis of proteins required by chloroplast, such as Rubisco, ATP synthase, etc. So guys, we have written 4 points over here. We can also write more points as I discussed earlier, right? Now let's move on to part B, outline the main features of cyclic photophosphorylation. So guys, cyclic photophosphorylation is one of the reactions that occur in the light dependent stage. So cyclic photophosphorylation is a part of light dependent stage. If we quickly recall cyclic photophosphorylation, so what happens during cyclic photophosphorylation? I have simply drawn a mini diagram for you to explain cyclic photophosphorylation. You need to know that in cyclic photophosphorylation, photosystem 1 is involved. And how does the cyclic photophosphorylation begins? Light from different wavelengths of light, or we can say light energy from different wavelengths of light, is absorbed by accessory pigments. So guys, what are these? These pigments are accessory pigments in the photosystem 1, and this one is the primary pigment or chlorophyll A, right? So what happens is that accessory pigments, what they are doing is that these accessory pigments are absorbing different wavelengths of light. And what accessory pigments do after absorbing different wavelengths of light? All the energy that is absorbed by accessory pigments is passed on to the primary pigment or chlorophyll A. You need to know that the primary pigment that I have drawn with light green over here is chlorophyll A, right? So this is chlorophyll A, which is the primary pigment. So light energy from different wavelengths of light is absorbed by accessory pigments, and then the energy is passed on to the primary pigment or reaction center. Because of this, electrons are excited and they are removed from the chlorophyll A or from the photosystem 1, right? Electrons are being removed from chlorophyll A of photosystem 1. These electrons are then picked up by what? These electrons are then picked up by electron transport chain, and then the electrons flow along the electron transport chain from one carrier to the next. As the electrons are flowing through the electron transport chain, what happens? Energy is released to pump hydrogen ions. From where? From stroma to the thylakoid space. So guys, this is the thylakoid membrane, and as I just discussed, the thylakoid membranes or the membranes of the grana contain photosystems. They also contain electron transport chain. They also contain ATP synthase, right? So this is the electron transport chain. This is ATP synthase. All are found within the thylakoid membranes. So we were on the flow of electrons along the electron transport chain, and as the electrons are flowing along the electron transport chain, energy is being released, and this energy is basically used to do what? To pump hydrogen ions. From where? From the stroma to the thylakoid space, the space inside the thylakoid. You need to remember that thylakoid space has a higher concentration of hydrogen ions, right? So hydrogen ions are basically pumped from a lower concentration to higher concentration. Eventually, the concentration of hydrogen ions increases in the thylakoid space. It was high already, now it has greatly increased. Because of this, a proton gradient is established between the thylakoid space and the stroma. So these hydrogen ions then return back from the stroma to from the thylakoid space to the stroma via the ATP synthase enzyme. As these hydrogen ions flow down the gradient, down the concentration gradient from thylakoid space to stroma, energy is released to make ATP from ADP and inorganic phosphate. This is what is cyclic photophosphorylation. Eventually the electrons from the electron transport chain will return back to the primary pigment of photosystem 1 or chlorophyll A of photosystem 1. Let's write this down. Light energy is absorbed by accessory pigments of photosystem 1 or PS1, and the light energy is then passed on, or we can say, and the energy is passed on to the primary pigment or chlorophyll A. This causes excitation of electrons which are then lost from chlorophyll A. What's next? Electrons are then picked up by the electron transport chain and electrons flow along the electron transport chain. Electrons flow along the electron transport chain, releasing energy to pump hydrogen ions or protons from stroma to thylakoid space. Then proton gradient is set up, or a hydrogen ion gradient is set up across thylakoid membrane. So protons diffuse back from thylakoid space to stroma, releasing energy to make ATP by chemiosmosis. Right? This process of ATP formation over here is called chemiosmosis. Basically, chemiosmosis involves the diffusion of hydrogen ions down the concentration gradient from thylakoid space to stroma, and this releases energy to make ATP. This is called chemiosmosis. So we are done with part B. Now we will move on to part C. Let's discuss part C. Red algae are aquatic prototists that are multicellular. The cells of the red algae have chloroplasts containing photosynthetic pigments. Many red algae live in deep water. Two of the accessory pigments of red algae chloroplasts are phycoerythrin, which appears red, often present in large quantities, and phycocyanin that appears blue. The first few meters of water nearest the surface absorbs the red wavelength of light. If the water also contains particles of organic material, it absorbs blue wavelengths of light. So, guys, what they are trying to say in this information is that the red algae live in the deep water. So, for example, this is the layer of red algae in deep water, right? And they're trying to say that few meters nearest to the water surface. So this is the surface of the water, and few meters nearest to the surface of water, which wavelengths of light are absorbed? Red wavelengths and blue wavelengths. So if you guys recall, how many wavelengths of light are there? Or we can say visible light contains how many wavelengths? 7 wavelengths: violet, indigo, blue, green, yellow, orange, and red, right? So what happens is that red wavelength is absorbed at few meters of distance from the surface of water. So this means that red wavelength does not reach the red algae. Same goes for the orange wavelength. Why is that so? Because red and orange wavelengths come in the same region, right? All right. What about the blue wavelength? If organic material is present at the near the surface of water, what happens is that blue wavelength is also absorbed and does not reach red algae. Same goes for indigo and violet wavelengths. That's because violet and indigo wavelengths come in the violet blue region together with the blue wavelength. This means that only green light and yellow light reaches the red algae. So the red algae must absorb these lights to carry out photosynthesis, right? And the pigments should be such a way, the pigments present should be such a way that they absorb green and yellow lights maximally, right? Okay, let's further read the question. Figure 2.1 shows absorption spectra of some pigments in the red algae chloroplasts. So guys, we can see that red algae chloroplasts contain chlorophyll A, which is the primary pigment, phycoerythrin, and phycocyanin. So we can look at this graph over here. This graph shows the wavelength on the x-axis and percentage light absorption on the y-axis. So this is absorption spectrum, right? What we can see is that chlorophyll A, this firm line represents chlorophyll A. Chlorophyll A absorbs best in which region? In the violet blue region, right? And also in the red region. All right. What about the other pigments like phycoerythrin? Phycoerythrin is absorbing best in blue region, in green region, in yellow region, right? And phycocyanin is absorbing best in which region? It's absorbing best in red, or we can say yellow region. Not red region, yellow region. Red region starts from here. So let's recall. If we talk about chlorophyll A, this is chlorophyll A. Chlorophyll A is absorbing best in which region? In violet blue region, right? And also in red region begins here, right? So chlorophyll A is absorbing best in red region and in blue region, right? Blue violet region. What about this pigment? This is phycoerythrin. Phycoerythrin is absorbing best in which region? In the blue region, in the green region, in the yellow region as well. Right? And if we talk about phycocyanin, it is absorbing best in which region? It's absorbing best. Phycocyanin is absorbing best in yellow region. Red region is starting over here, right? All right. Let's have a look at the question. Describe the differences in the absorption spectra of the three photosynthetic pigments shown in figure 2.1 and explain how these differences help red algae to survive in deep water. So guys, what we have to do is that we have to describe the three absorption spectra that we just saw in this graph. So we have to describe the absorption spectrum of chlorophyll A, phycoerythrin and phycocyanin, and then we have to explain that how these absorption spectra help algae, or how these pigments found in the chloroplast of red algae help it to survive in deep water. So guys, let's first describe the graph or the absorption spectra. So we can say that chlorophyll A has high absorption at which wavelengths? We need to identify the wavelength, right? So if we talk about chlorophyll A, so chlorophyll A has high absorption at which wavelength? It's 425 nanometers, right? And what else? Which wavelength? 675 nanometers. So we can write that chlorophyll A has high absorbance at which wavelengths? 425 nanometers and 675 nanometers. Chlorophyll A has highest percentage light absorbance at which wavelength? 425 nanometers. Then we can talk about phycoerythrin, right? So phycoerythrin has high absorbance at three different wavelengths. And what are those wavelengths? One is 455 nanometers. Other one is 520 nanometers. And the third one is 565 nanometers, right? If we talk about phycocyanin, phycocyanin has high percentage absorbance at which wavelength? It has high percentage absorbance at 610 nanometers, right? All right. So let's mention these values because this is how we describe the absorption spectra. We basically identify the wavelengths for high absorbance by different pigments. So let's write about chlorophyll A first. What we can write is that chlorophyll A has high absorbance or high percentage absorption, high percentage light absorption at which wavelengths? 425 and 675. At 425 and 675 nanometers. All right. let's talk about phycoerythrin now. Phycoerythrin has high percentage light absorption at 455, 520 and 565. All right, so phycoerythrin has high percentage light absorption at 455, 520, at 455 nanometer, at 455, 520 and 565 nanometers. Then we talk about phycocyanin. Phycocyanin has high percentage light absorption at which wavelength? At 610 nanometers. All right. So these are the points for description. We get 3 marks out of 7 by describing the graph, right? Chlorophyll A, phycoerythrin, and phycocyanin. Now what we have to do is that we have to explain that how these differences help red algae to survive in deep water. So guys, as we discussed earlier, which lights are reaching the red algae? Only yellow and green lights. And if we look at the absorption spectrum or the absorption spectra, so green and yellow lights are mainly absorbed by which pigments? Phycoerythrin and phycocyanin, right? Green and yellow light is reaching red algae and it's mainly absorbed by phycocyanin and phycoerythrin. Chlorophyll A is not absorbing these lights. So what we need to mention? We need to mention that chlorophyll A has we can first we can write that only green and yellow light reaches red algae in deep water. Then we can write the chlorophyll A has little or no percentage light absorption of green and yellow light. However, phycoerythrin and phycocyanin have high percentage light absorption of, or we can write in yellow-green region and yellow-green region. So guys, we can see that the red algae is surviving in deep water because of these two accessory pigments, phycoerythrin and phycocyanin, right? So you can mention that, however, phycoerythrin and phycocyanin accessory pigments have high light absorption or high percentage light absorption in yellow-green region. These accessory pigments are absorbing light which is not absorbed by chlorophyll A, which is the primary pigment, right? And just because of these pigments, red algae are surviving, and they have high growth in deep water. So what we can say is that light absorbance allow red algae to carry out light dependent stage, because light is used in light dependent stage. Light is used in light dependent stage, right? You can mention that photolysis and photophosphorylation occurs. Producing ATP and reduced NADP, producing ATP and reduced NADP, which are used in Calvin cycle to produce carbohydrates and other nutrients which allows the red algae to survive in deep water, to grow and survive in deep water. So that's it for 7 marks. We have right sufficient points. I hope this question is clear to all of you. Now we will move on to the next question.